The nature of prokaryotes and eukaryotic genes. When Gregor Mendel put forward his laws of inheritance in 1860, he used the term hereditary factors to explain what control characters shown in a phenotype and which are transmitted from generation to generation. At that time, these were imaginary units where they were located in cellular structure was not known. These physical and functional units of heredity have been identified as genes that are located on chromosomes as discrete units. This disclosure began with the development of cytology and the ability to observe the behavior of chromosomes in mitosis and meiosis. Behavior of chromosome and the behavior of the Mendel's hereditary factors show the same pattern. In eukaryotes, chromosomes occurs in pairs in diploid somatic cells, and hence genes also occurs in pairs. A pair of chromosomes coming from the two parents containing the same gene is called homologous chromosome. Generally, prokaryotes have one chromosome in each cell and hence may be considered haploid. The location or the site of a gene on the chromosome is called locus, loci in plural. The alteration version of genes that are located at the same locus on different chromosomes are called alleles of the gene. Prokaryotic genes are housed in loci on the circular DNA molecule as discrete segments of DNA. There are many steps in biochemical pathway. Each step is controlled by a gene. Therefore, many genes involved in controlling a particular phenotype. In eukaryotes, these genes are scattered among several chromosomes. While in prokaryotes, they are arranged in the same region of the chromosome, one after the other, as clusters. These clusters are expressed together by a single control region and are transcribed into one mRNA. Such mRNA molecules will be translated into one mRNA. Such mRNA molecules will be translated into several different peptides. These organized gene clusters in prokaryotes are called operands. In prokaryotes, all DNA segments in the chromosome are functional, either transcribed into mRNA or act as control region. Why in eukaryotes, a large portion of DNA has no identified functions. Such DNA segments that are present in between the genes are referred to as intergenic DNA. There are also sequences with the genes that are transcribed but are not translated into polypeptides. That means there are coding sequence as well as non-coding sequence in the transcript of the gene. The non-coding sequence with the genes are called introns, while the sequence that code for polypeptides are exons. Accordingly, the transcript contains exons as well as introns. The transcript is a pre-mRNA and it undergoes processing where introns are excised. Exons are joined in order to produce mRNA. Gene is the fundamental physical and functional unit of heredity. A gene is comprised of a segment of DNA on a specific site on a chromosome. It specifies the sequence of RNA. Operin, a group of genes that function as single transcription. It comprised of a control region, an operator, a promoter. Structural genes that are transcribed into one mRNA, coding for several peptides.